document published in 2019, titled Broad-Spectrum Coronavirus Antiviral Drug Discovery, clearly states that Fort Detrick did use reverse genetic systems to bioengineer pre-emergent bat coronaviruses. Reverse genetic systems advances in the study of highly pathogenic coronaviruses and potential pan-coronavirus drug candidates partially depend on the technology to genetically manipulate COVs to probe mechanisms of viral pathogenesis and antiviral drug activity. Reverse genetic systems synthetically generate viruses from known viral sequences. In situations where clinical isolates of infectious material are unavailable due to restriction for collecting patient samples, shipping infectious materials, or availability of containment laboratories, reverse genetic systems provide essential research materials for studies on viral pathogenesis and model development. Prior to the SARS pandemic, robust reverse genetic systems to manipulate the genomes of COVs had already been developed by systematic assembly of knockasets into full-length infectious clones, allowing precise and targeted genetic manipulation of viral genes. Infectious clones allow the creation of near-homogeneous viral stocks, whereas traditional viral stocks are prepared by amplification of infectious material in cell culture over many passages. Strategies to build reverse genetic systems were rapidly applied to both SARS-CoV and MERS-CoV within the first year of identification of these viruses. In addition to reconstructing epidemic strains of COVs, reverse genetic systems allow targeting of mutations to specific viral genes and assembly of viruses when infectious material is not available. As an example, the ability to isolate mutations in particular genes was applied to studies of the spike S glycoprotein of SARS-CoV, while maintaining the isogenic background of the viral replicase and other structural proteins. Mutations from zoonotic, early, middle, and late epidemic strains of the SARS-CoV outbreak were inserted into the S glycoprotein of the epidemic strain of SARS-CoV, Urbani, to determine the effect of evolution on viral entry into human cells, as well as viral pathogenesis in rodent and primate models of disease. By targeting mutations to a specific viral gene, reverse genetic systems allow researchers to probe cause and effect relationships of host pathogenic responses to viral genetic changes. In addition, reverse genetics techniques were utilized to study pre-emergent BATCoV strains. Recombinant versions of BATCoV HKU3, BATCoV WIV1, and BATCoV SHC014, SARS-CoV-like, as well as BATCoV HKU5, MERS-CoV-like viruses, were generated and used for in vitro and in vivo models of emerging coronavirus disease. Panels of zoonotic, epidemic, and pre-emergent viruses synthesized by reverse genetics techniques encompass a diverse array for use in high-throughput platforms for the discovery of countermeasures that are effective against the broadest range of COVs without being reliant on procuring clinical isolates.